Welcome in, and thanks for grabbing a seat at the table with us today.
Absolutely. It's great to be here.
If you could see our setup right now, you'd notice we've um, completely transformed the space for you. The backdrop behind us looks like this neon-lit, futuristic laboratory that just sort of seamlessly melts into a massive humming server room.
It's a pretty striking visual.
Yeah, we set this up because it's the absolute perfect visual for the fusion of biology and coding we're about to explore. Today, you and I are going on a deep dive into a structural transformation in medicine called Pharmaceutical Superintelligence, or PSI.
Which is a heavy title, but it
It
fits.
really does. Our mission today is to explore this, well, it's an audacious reality that's unfolding right now in the scientific community. We're talking about taking a simple, plain language request, like, typing a sentence into a terminal, and initiating a fully autonomous end-to-end drug development program. Literally, prompt to drug.
I know it sounds like an ambitious science fiction pitch, but the underlying architecture is already functioning.
Right, and to guide us through how this is actually possible, we've got a really fascinating stack of sources today.
Yeah.
We're looking at two major technical papers. One is on automated drug development multi-agent processes, and the other is titled Orchestrating Pharmaceutical Superintelligence.
Both are incredibly dense, but very revealing.
Extremely dense.
Hmm.
But before we get into the actual machinery of it all, let's just set the stakes. Traditional drug development is a notoriously brutal pipeline. We are looking at timelines extending well over a decade, with a nearly 90% failure rate once these drugs actually hit the clinical stages.
Yeah, 90%.
I mean, that is 10 years of work and billions of dollars, only to face a 90% chance that the drug won't ever make it to the pharmacy shelf.
And those failures, they typically originate at the very beginning of the research phase.
How so?
Well, human limitations in integrating massive data sets, or simply an inability to predict how complex biomolecules will interact in a human body, it leads scientists to prioritize the wrong compounds early on. What we are analyzing today is a direct solution to that fragmentation.
Right.
The industry is basically moving away from human trial and error and replacing it with a system of systems. We have to start imagining AI agents not just as, passive software tools, but as active co-scientists.
Okay, let's unpack this system of systems concept, starting with what the sources refer to as the orchestration layer. Instead of thinking about this like a simple software program, it helps to look at it through the lens of systems engineering, like a corporate hierarchy almost.
That's a great way to visualize it.
At the top level, you have the user level. This is where the human scientist enters a natural language prompt, establishing the core mission objective for the AI. Below that, the architecture splits into a cohort level.
Right, the departments.
Exactly. Distinct modular groupings, like a biology cohort, a chemistry cohort, clinical planning, process engineering. And finally, down at the granular level, you have the agent level. These are highly specialized digital workers executing discrete technical tasks, whether that means running a thermodynamic simulation or firing up a physical liquid handler in a lab.
What's fascinating here is how these agents communicate across those different levels.
Because they actually talk to each other now.
Yes. In older automation models, you essentially had static scripts running in isolation. A program did its job, handed over a file, and stopped. But in this pharmaceutical superintelligence framework, the agents use highly robust communication protocols.
Like Google's A2A or Anthropic's MCP.
Precisely. This allows them to maintain a shared context and a continuous memory across the entire development lifecycle.
But in standard pharmaceutical development, the early-stage molecular designers rarely talk to the late-stage manufacturing teams until years down the line. It's totally siloed. How does this shared memory protocol actually change that dynamic?
It completely dismantles the silos. Because the memory is shared across all cohorts instantaneously, an insight derived from a late-stage manufacturing simulation can dynamically inform how the chemistry cohort designs the initial molecule on day one.
That's incredible.
It is. The system knows that a specific chemical bond might be too unstable to manufacture at an industrial scale, so it prevents the early-stage AI from even proposing that structure.
That is such a massive shift in efficiency. Let's look at the very first step of this pipeline, picking the target. Because if you pick the wrong biological target to attack, no amount of brilliant chemistry is going to save the drug.
Very true.
The sources highlight a specific system from the biology cohort called Pandiomics Target Identification Pro, or TID Pro.
And this is where the machine learning models get incredibly sophisticated. TID Pro utilizes a positive, unlabeled learning framework.
Which means what exactly?
Well, in biology, we actually have very few perfectly validated, universally agreed upon drug targets. So instead of getting confused by the vast unknowns, the system treats the known successful targets as positive and literally every other gene in the human body as an unlabeled candidate.
Okay, so it's not guessing. It's comparing against a massive unlabeled pool.
Right. And to evaluate them, it analyzes 22 distinct features. 12 of those are based on omics data, which involves mapping total biological systems like genomics or proteomics, and the other 10 are driven by extracting context from scientific text.
Looking through the sources, I notice it doesn't apply the same weight to every disease. The text mentions a SHAP analysis. How does the system adapt its search depending on what kind of drug you asked it to build?
The SHAP analysis reveals that the AI understands context is everything. If the initial prompt asks for an oncology target to fight cancer, the system relies heavily on matrix factorization.
Which is?
It's a mathematical way of breaking down massive grids of genetic relationships to find hidden patterns. But if the prompt asks for a metabolic or neurological target, the system shifts its weight. It starts relying much more heavily on attention scores derived from reading vast oceans of published scientific literature to find connections human researchers might have missed.
I want to share the performance stats on this for you, because the gap between this and standard AI is wild. When tested on a benchmark of 38 different diseases, general frontier large language models, the really advanced chatbots you might use for daily tasks, they only retrieve the correct clinical targets between 15 and 40% of the time.
Which is pretty poor.
Yeah, not great. But TID Pro hit a 71.6% retrieval rate. It is prioritizing what the sources call actionable intelligence. When TID Pro finds a novel target, there are typically over 500 available bioassays for it. The general models only find targets with around 350.
So it doesn't just find the theoretical answer, it finds an answer that human and robotic scientists can actually test in a lab today. This raises an important question, though. Once you have that perfect biological target, how do you build the therapeutic molecule to interact with it?
Because standard models kind of struggle there.
Exactly. Standard one-dimensional modeling approaches often just memorize past structures and really struggle to explore entirely new chemical space.
Here's where it gets really interesting. We pivot over to the chemistry cohort, and they utilize a framework called MUL-IT. This uses hierarchical discrete diffusion models, or HDDM.
The HDDM approach is brilliant.
The best way to visualize this process is to think of a sculptor working with a block of clay. The HDDM establishes the high-level shape first. It decides if a section should be an aromatic group, like a ring of atoms, or an aliphatic group, which is more of a straight chain of carbon. Only after the broad three-dimensional shape is secured does it carve out the specific atomic details.
It also solves a major historical bottleneck in AI drug design by using decoupled atom encoding.
Right, because older models used to mess up the geometry.
They did. In older models, the AI would frequently get confused by complex chemical motifs. A great example is the pyrrolic nitrogen motif.
Sounds complicated.
It's basically a specific five-sided ring of atoms containing nitrogen that is notoriously tricky for algorithms to map out in 3D space. The older AI would try to assign multiple physical states to a single token, leading to systematic reconstruction failures. MUL-H explicitly represents aromaticity and charge for every single atom, which stops those structural failures from happening.
So the system has identified the target, and it has generated structurally sound molecules. The next hurdle is figuring out if they actually bind together. The sources introduce the Isomorphic Labs drug design engine, or ISO-DE. The text notes it more than doubles the accuracy of AlphaFold 3 on really challenging benchmarks, like out-of-distribution events.
And an out-of-distribution event is just something the AI hasn't explicitly seen in its training data.
Like a totally new target.
Yes. For example, the sudden opening of a novel, cryptic pocket on a protein surface. These are hidden crevices that only appear when the protein shifts its shape. Previously, you could only discover these pockets through painstaking experimental crystal soaking in a physical lab. ISO-DE can actually model these hidden pockets digitally.
The performance metric they use to measure this binding affinity prediction is the mean Pearson correlation, represented by the letter R. Looking at the sources, traditional physics-based methods, specifically the gold standard they call FEP+, they hit a correlation of 0.78. But ISO-DE hit 0.85. How is a deep learning model beating a heavy, computationally expensive physics model like FEP plus R?
It really comes down to active learning and iteration. Deep learning models can synthesize vast amounts of structural data and recognize binding patterns that pure physics simulations take weeks to calculate. Because it's deep learning, the computational overhead is drastically lower.
That's faster.
Much faster. And the AI doesn't just make a prediction and stop, it actively learns. The sources detail a workflow called ChemScreener, which explores new chemistry continuously. In a real-world case study targeting a specific cancer-related site called the WDR5W-WIN site, this active learning approach bumped the hit rate from a baseline of 0.49% up to 5.91%. Yeah, that represents a massive leap in identifying completely novel molecular scaffolds that could become blockbuster drugs.
That optimization is impressive, but standard models are built to be good on average. What happens when the AI encounters a highly specific mutant protein or a rare virus that doesn't follow normal structural rules?
That is where the system employs protein test time training, or protein TTT. This protocol allows the AI to customize itself to a specific protein on the fly.
On the fly.
Exactly. It operates on a mathematical concept called perplexity. By minimizing its perplexity, which is essentially reducing the AI's surprise at the protein's unique, unusual patterns, it updates its own parameters to generate much more accurate representations of that specific target.
The results of that customization are striking. On something called the Big Fantastic Virus Database, standard models failed to predict the structures of many viral proteins. But when they applied protein TTT, those structure predictions improved for 19% of those previously unsolvable targets.
Which is a huge unlock for virology.
Totally. But reading through all this generative AI capability, I kept wondering about hallucinations. If a language model hallucinates a fake historical fact, it's annoying. If this AI confidently designs a molecule that is physically impossible to synthesize, it derails the whole multi-million-dollar process.
Right, and the stakes are much higher. To ensure structural integrity and prevent those hallucinations, the framework integrates a safeguard called the confide system. It measures what the researchers call topological frustration.
Topological frustration.
It looks deeply into the latent representations of the molecule, which are the underlying mathematical fingerprints the AI uses to understand the structure. By analyzing those fingerprints in an unsupervised manner, the system detects irrational geometric arrangements before they are ever proposed. The sources note the system represents a 73.8% relative improvement over standard confidence metrics.
So the digital orchestration is operating perfectly. The AI found the target, designed the molecule, verified the binding, and double-checked the physics to ensure it actually exists. But a digital molecule living on a server doesn't cure anyone. How does this system transition into the physical world?
This brings us to the physical lab.
Yes, the DMTA cycle design, make, test, analyze.
This is where we enter the realm of self-driving labs. These are high-throughput facilities where the robotic execution layer takes over. The orchestration in the physical space is handled by systems like ChemAgents.
ChemAgents has its own modular structure. You have a task manager agent that coordinates 4 distinct roles. There is a literature reader that ingests synthesis protocols, an experiment designer that lays out the step-by-step chemical reactions, a computation performer analyzing the physical data inline, and a robot operator that actually manipulates the hardware.
And we are talking about highly advanced hardware.
Right. Modular robots with 360-degree arms, thermal-controlled liquid handlers, and split-cool bead dispensers.
However, there has always been a major hurdle in laboratory automation, often called the legacy equipment gap.
I found this part fascinating.
It is. Most scientific instruments, centrifuges, incubators, spectrometers were designed over the last 50 years to be operated by human hands. Tasks like changing a reagent bottle or opening a latch require human dexterity, which traditionally shatters the dream of a fully autonomous 24/7 lab.
The solution to that gap is wild. Because they couldn't practically redesign every piece of legacy equipment on Earth, they introduced bipedal humanoid AI scientists.
Literal humanoids.
Yeah, the example cited in the text is in silico medicine supervisor. This is a walking bipedal robot that physically navigates the laboratory. It pipettes samples, interacts with older legacy equipment, and works around the clock alongside human scientists.
And that humanoid acts as just another agent within the central orchestration framework. If we connect this to the bigger picture, this closed-loop system, where the AI designs the experiment, the robots execute it, and the physical data feeds instantly back into the AI to adapt the very next experiment, results in a massive compression of time.
The numbers definitely back that up. For you listening, the sources state that this closed-loop system compresses the timeline from project initiation to nominating a preclinical candidate from 4.5 years down to just 12 to 18 months.
But once that humanoid robot pipettes the winning compound, you suddenly face an entirely new set of problems. A lab can make one perfect pill, but a global patient population needs millions of them. How does the system handle industrial scale-up without the manufacturing costs bankrupting the company?
This is orchestrated by the Superintelligence system. It's structured into 3 specific cohorts: knowledge, concept, and parameter. The knowledge cohort acts as the foundation, pulling technical literature and turning it into semantic graphs so the AI understands real-world process constraints. Then the concept cohort steps in to design the factory floor,
And they utilize an adversarial design critique interaction, which is really clever.
basically arguing with each other.
Exactly. One agent proposes a process flow diagram for the chemical plant, and a correction agent acting essentially like a strict veteran process auditor tries to find topological flaws or chemical engineering violations. It actively looks for reasons why the plant design will fail, forcing the system to refine the blueprint before it ever reaches the simulation phase.
And the parameter cohort.
Finally, the parameter cohort interfaces with industry standard solvers to optimize the specific factory settings, like reactor temperatures, residence times, and reflux ratios.
The return on investment for that adversarial collaboration is massive. By optimizing those factory parameters, they cut operating expenses for a process called butadiene hydrocyanation by 33%. For aromatics purification, they saw a 21% cost reduction while still maintaining a 97.2% purity level.
They are saving tens of millions of dollars before a single machine is turned on.
But having a cheap, pure drug doesn't mean it's approved. It still has to survive clinical trials. The orchestrator shifts gears to clinical development modules, using engines like InClinico to forecast the probability of success for Phase 2 trials.
Setting up the trial itself also gets automated. A tool called AutoTrial scrapes data from global registries and extracts PICO data, which stands for Population, Intervention, Comparator, and Outcome, achieving a 97% classification accuracy. But the most profound shift in the clinical phase is the implementation of digital twins.
I understand the concept of a digital twin for a jet engine or a chemical plant, but how does this system use a digital twin for a human patient in a medical trial?
It uses deep learning to simulate a highly accurate trajectory of how a specific patient's disease would progress if they received no treatment.
Like a baseline.
Exactly. By generating these digital twins, researchers can adjust for baseline covariates. The digital twin effectively acts as a simulated control arm for the trial. You can compare the actual patient receiving the new drug against their own simulated digital twin.
Which means you need significantly fewer actual human beings enrolled just to receive a placebo.
Right. You achieve enhanced statistical precision while exposing far fewer patients to non-active treatments.
That has massive ethical and financial implications for trial design. But anyone who works in this industry knows the absolute worst bottleneck isn't always the science.
Oh, it's the red tape.
The regulatory paperwork. To get a drug into a human, you have to submit investigational new drug dossiers to regulatory bodies like the FDA. It is an immense mountain of highly technical writing.
And AI agents are obliterating that administrative bottleneck. Systems like AutoIND draft these massive summaries directly from the raw source documents.
When I saw that an AI was writing regulatory dossiers, I initially worried about the system hallucinating data to make the drug look better. But the sources highlight a mechanism called retrieval augmented generation, or RAG. What is that mechanism actually doing to ensure accuracy?
RA is a crucial safeguard. It means the AI isn't just writing paragraphs based on its own parametric memory, which is how standard chatbots often hallucinate facts. Instead, RAGACA forces the AI to actively retrieve the actual raw clinical trial data, cite the specific sentences from the source documents, and generate the report based exclusively on those grounded facts. It ties the generated text directly to verifiable data.
The comparative efficiency stats on this are staggering. For an experienced human medical writer, it takes about 100 hours to draft a non-clinical summary. The AutoIND system completes it in 3 to 4 hours.
But that is a 97% reduction in time.
Manual human output is generally 1 to 2 pages a day. The AI generates 50 pages an hour with no critical errors. The system even deploys regulatory intelligence agents that constantly scan global agencies, so the paperwork is proactively compliant with any new rules that drop that morning.
Which brings us to the final, crucial component, quality governance. You cannot let an autonomous system run a pharmaceutical pipeline without rigorous oversight. They employ a frontier QA model. It's an entire army of agents dedicated solely to compliance. They ensure every action adheres to ALCOA plus principles.
For those outside the compliance world, ALCOA Plus means every piece of data must be attributable, legible, contemporaneous, original, and accurate.
Right. They deploy specific agents for generating requirements, writing test scripts, and autonomously conducting vendor audits. Out on the factory floor, machine learning models continuously monitor equipment performance. They can predict machine failures with 70% accuracy.
That's huge for uptime.
Very huge. That predictive maintenance capability alone cuts maintenance costs by 45%. It provides continuous, real-time risk management across the entire ecosystem.
Okay, let's unpack this. So what does this all mean? Let's bring this home for you. The core takeaway from this deep dive into these sources is that we are officially crossing the threshold into a scientific intelligence paradigm. AI agents are no longer just sophisticated calculators we use to check our math.
No, they are active co-scientists.
They are collaborating with bipedal humanoid robots to design, synthesize, test, and scale life-saving therapies at speeds that were literally impossible 5 years ago.
This raises an important question, though, and it's something I want to leave you to mull over. If we truly achieve this prompt-to-drug reality, if an orchestrated system of AI agents and humanoids can autonomously handle everything from the initial biological spark of an idea through the clinical trials all the way to the final factory output, what becomes the primary role of the human scientist?
That is the existential question at the center of all this.
In a world where the execution of complex science is perfectly and autonomously automated, perhaps the ultimate human skill will no longer be finding the answers. The orchestrated machines will find the answers faster and more accurately than we ever could. The true art, the highest value of the human mind going forward, will be learning how to engineer the perfect prompts to ask the right questions.
From answer finders to question askers, that is an incredible paradigm shift to think about. Well, that is all the time we have for today. Thank you so much for grabbing a seat at the table and taking this deep dive with us. Stay insanely curious, and we'll see you next time.